Hi, I'm Alyssa Weinstein, the National Seed Bank Manager, and behind me here is the brand new National Seed Bank facility. Our mission is to safeguard Australia's flora for future generations. So we do that by collecting seed from wild populations and keeping it safe here in our vault, as well as developing protocols to germinate these seeds. We look at species from the ACT region, particular focus on alpine and subalpine ecosystems, as well as species from across Commonwealth parks, so Uluṟu, Kakadu, Booderee, Norfolk, Cocos and Christmas. We also work with traditional owners to develop target species of plants that are culturally important to them. Behind me you can see the area where the field cars come in from collection trips, and this is really a beginning of a seeds journey into the seed bank. After that, the seeds are taken through into the facility and into the dry room, where I'll take you now. We're standing outside the dry room, and this is where our seed collections are initially stored once they come in from the field. And they stay in the dry room until they're ready to go into the vault. Throughout that time, data is collected about the seeds. Before they go into the vault, they also need to desiccate. So the room is kept at 15 degrees Celsius and 15% humidity, and that ensures that the seeds are dried down. One of the seeds here is from Uluṟu-Kata Tjuṯa. This is actually the rarest plant in the ACT and a major conservation focus for us. There's less than 10 individuals left in the wild which haven't been setting seed. And here at the National Seed Bank, we've been working with the Conservation Horticulture team, and we've actually managed to give germinated seeds to the Conservation Horticulture team. And these seeds have then been grown up into adult plants. And through this, we've effectively doubled the population size of this threatened species.
My name's Gemma Hoyle, and I'm a seed scientist here at the National Seed Bank, so I carry out a lot of research on the collections. We're currently in the seed prep laboratory. This is where the collections come fresh from the field to be cleaned and processed. Often they come in very bulky, and they include plant material and fruits and flower parts, and all that needs to be removed from the collection. So often collections go from being very big and bulky down to very small, and that means that we can bank more in the vault. This stage is really important because it increases the quality of the collections that we put in the bank and the quality of the collections that ultimately we take out of the bank and do research on and grow plants for translocation, etc. So we need to be really careful in this work that we're not throwing away precious viable seed, but that we are discarding all the chaff and plant material that we don't want to put in the bank that isn't seed. So this is the X-ray room. So we X-ray a small proportion of each collection, and we calculate the percentage seed fill. We're looking for seeds that have an embryo inside that isn't damaged, it's whole and it's mature, fully formed embryo. This is the seed morphology room. This is where we image every collection. So what we try and do is image a group of seeds per collection so that we can see that diversity within the species. And then we also do seed dissections. And what we're looking at is the internal morphology of each species.
So we're standing in front of the seed storage vault, and this is really the heart of the National Seed Bank. This is where all of our collections are stored. So we have over 8,000 accessions or collections, and that's of over 4,000 species, and almost 200 of those are listed under our national environmental legislation. Seeds are stored in the vault to extend their longevity, and part of our research program here is testing the seeds and determining their lifespan in the vault and optimising storage protocols. And there's actually quite a bit of variability in species and the storage conditions that different species require, and how to best keep them safe is going to be very different for one species than from another. So here are our seed collections, and what you'll notice is we've got much more room to expand. So in our old facility, we were at capacity with these collections in the vault, and now we've got almost quadruple the space to expand our collections. That means more species brought into the vault, greater genetic diversity captured within those species as well. So this is our collections, and you'll see in here there's also some silica gel, and that helps with the humidity control.
So there's no point having seeds in the seed bank if we don't know how to germinate them. And the germination requirements of each species are very different. We need to know how to use the seeds. We need to be able to assess the seeds periodically to check that they're still alive. And we need to be able to use the seeds in seed science research and in the future when we want to use them for translocation and restoration purposes. So this is the seed biology lab. This is where we carry out all the research into the collections, and primarily we're looking to uncover the germination requirements of each species. I guess I inherit all the problem collections, the species that haven't germinated in the initial germination tests will come to me. And then, it's exciting to try and mimic the natural environment, think about the ecology of the species and recreate that in the laboratory. But yeah, once we do crack the code and we start to see germination in the incubators, it's very exciting. And sometimes we're seeing seedlings of a species for the first time. So this is the seed bank incubator room. This is where all the germination tests are brought and they're put at different temperature regimes. So, for example, collections that come from Kakadu, we've got a nice warm Kakadu temperature regime. We've got an alpine spring regime. We've got a grasslands growing season regime. So depending on where the collections come in from, they'll go into the appropriate germination incubator. We sow seeds onto agar, which keeps the seeds hydrated, and it also allows us to see the early signs of germination. So if seeds were in potting mix, for example, we wouldn't be able to see the germination happening. There's so much potential in each experiment that I set up. I'm always aiming to fill a gap in the knowledge, and so we're always investigating things that haven't been studied before, and then there's always that potential that you'll have the scientific breakthrough.
So the National Seed Bank is within the Australian National Botanic Gardens, and that's really helpful as it enables us to partner directly with the National Botanic Gardens Conservation Horticulture team and grow plants from seeds into adults and get them to be part of the living collection. So here behind me you can see some plantings that have come out of the National Seed Bank Research Program. We're also really fortunate to be directly opposite ANU across the road, which is really helpful to facilitate research collaborations. So I originally did my PhD at ANU in plant pollination ecology, and through that work I was exposed to a big array of really unique Australian ecosystems. For me, that sort of sparked the realisation of these ecosystems really are threatened and I wanted to support their conservation. So for me, it's the perfect combination of research, science communication, but also being part of the broader biodiversity conservation agenda that we have nationally and internationally. Thanks so much for your interest in the National Seed Bank. It's been great to show you around today and look forward to catching you in the garden soon.